My name is Andreas Geier. I'm professor of internal medicine and hepatology at Würzburg University Hospital, Germany. The research focus of my laboratory is metabolic liver disease, but I also have a strong personal interest in anthropology and human history. This brought me in touch with Svante Pääbo, who has received the Nobel Prize in Physiology or Medicine in 2022 for his discoveries concerning the genomes of extinct hominins and human evolution. I would like to tell you today about the study that we have undertaken together with archaeogenetic researchers from the Max Planck Institute in Leipzig, Germany. The liver is the central organ in the human body responsible for the metabolism of carbohydrates and lipids. It plays a key role in fat storage under conditions of overnutrition, a condition termed metabolic dysfunction-associated steatotic liver disease, or abbreviated MUSCLE-D. This disease affects up to 30% of modern populations. Fat deposition in the liver is not only modulated by environmental factors, such as overnutrition, but also by genetic predisposition. What is known about genetics in liver steatosis? Here, Professor Marcin Krawczyk from Homburg University Hospital has been collaborating with us for a long time.
Thank you, Andreas. Indeed, in the last 2 decades, we've learned really a lot about the genetic background of hepatic steatosis. And the major genetic risk factor for fatty liver is localized in the gene called PNPLA3. Carriers of the minor allele of this variant are at risk not only of hepatic steatosis, but they also can develop liver fibrosis, cirrhosis, and HCC. Approximately 50% of Europeans with fatty liver carry this variant, but we also know that there are other parts of the world, like Mesoamerica, where it's even more frequent. Now, there is a question. How come a genetic variant which is associated with progressive liver disease is that common in our DNA? And one of the hypotheses that might explain it is called the thrifty gene hypothesis, which tells us that genetic variants which were advantageous during the late Paleolithic era are now harmful. Particularly, the ability to store fat was likely an advantage throughout most of the human history, while under modern day living conditions, it is detrimental.
In the past decade, the pioneering research by Professor Pääbo opened the new research field of archaeogenetics. His research enabled the first sequencing of the genome of the Neanderthal, the well-known extinct relative of present-day humans. Together with his former PhD student, Johannes Krause, now director of an own Max Planck Institute in Leipzig, Germany, he also discovered Denisova, a previously unknown hominin solely based on the remnant DNA of tiny little finger bones. Advances in ancient DNA technology now enable to study the flow and natural selection of genes by analyzing samples from populations throughout time and space over the last 50,000 years. In this study, we were interested to detect the ancestral origin of the PNPLA3 fatty liver variant and to understand the past trajectory of this variant throughout human evolution and history, and finally, to detect potential signals for selection over time, which could be explained by the thrifty gene hypothesis. This is where the experts from Max Planck Institute come into play. Dr. Stefan Schiffels will now explain to you the methodology.
Archaeogenetics is a method of extracting genetic data from human remains like teeth or bones from archaeological excavations. And we and other laboratories worldwide have, in the last 10 years or so, generated data from thousands of individuals who died hundreds or thousands or even 10,000 years ago, the oldest ones perhaps 40,000 years ago and older. And this data allows us to look directly into the past and see how allele frequencies change through time. For example, the frequency of the risk variant of PNPLA3. Of course, such changes could be due to natural selection. So people who carried a variant could have been fitter or healthier or otherwise produced more offspring than people without the variant. But it could also have been the result of random chance, simply because some people have more offspring than others. So allele frequencies change over time, and we need to assess how significant that change is with respect to, for example, changes that we see in other variants that are not under natural selection.
Now, what have been our results? Analyzing the reference genome sequence of primates, which you can see on the left side of the panel, it became clear that all great apes, orangutan, gorilla, chimpanzee and bonobo, which have split from the ancestral primate lineage, are actually carrying the ancestral less risky gene variant. On the right side, you can see the human lineage with modern and archaic humans. All available Neanderthal, in total 21, and the Denisovan individuals, in total 2, one of them the worldwide unique Neanderthal Denisovan hybrid, carried the risk allele, suggesting fixation in the ancestor of all archaic humans. Modern humans, even in the oldest available samples, carry both the reference allele, which is called wild type, and the risk allele for fatty liver. These findings led to the conclusion that the principal fatty liver gene variant PNPLA3 must have developed before the split of the human lineage some 700,000 years ago.
So now that we know that the risk variant was 100% in Neanderthals and variable in humans today and in the past, of course the question is, what has happened exactly? When we look at the distribution of this variant in the last 15,000 years ago, which is shown here in red, we see that in fact its allele frequency has decreased a little bit in all regions of the world, and looking at it with different techniques, for example, grouping individuals into populations or looking at individuals separately. But when we compare these changes to the changes of other variants that we know have not been subject to natural selection in the genome, we see that PNPLA3 did not change significantly in the last 15,000 years, suggesting that natural selection was not at play to the level that we can measure and in the time period that we're looking at, so 15,000 years ago.
What can these archaeogenetic findings tell us about liver steatosis in ancient and modern humans? Our findings lead to the obvious question: did Neanderthals have fatty livers? Of course, we cannot say. But one can speculate that our observation underscores a putative advantage of hepatic fat storage in cold climate, and particularly for Neanderthals under Ice Age conditions.
Supportive of this hypothesis is the recent data from the Yakut population in the coldest northeast region of Russia, where the PNPLA3 variant allele is predominant and can be detected in almost 90% of the population.
As a consequence from the present study, functions of PNPLA3 on heat production outside the liver certainly deserve future investigation. Hopefully, we will learn more about the functions of this particular fatty liver gene variant on the human body outside the liver.
Does the absence of signal of natural selection in the archaeogenetic data ultimately argue against the hypothesis of natural selection during the Paleolithic era?
While our genome-wide analysis did not detect natural selection, at least not significantly in the last 10 to 15,000 years, of course natural selection could have been at play at older times or over longer time periods. So this would then not be something that we can assess with archaeogenetic data.
The most obvious question arising from this study is whether the PNPLA3 variant represents genetic heritage from Neanderthals.
There's also the question on whether this variant was contributed from Neanderthals to humans when we met Neanderthal ancestors about 45,000 years ago. But this does not seem to be the case. So only very, very few of the variants that we see today in humans could have been perhaps contributed from Neanderthals. The dominant factor must have been the case that PNPLA3, at least the variant that we're looking at, is an old variant that is already in the human lineage since our time of the common ancestor between Neanderthals and modern humans.